So this is the first chapter of Unit 3. Okay, so amino acids and proteins. So amino acids are carboxylamines. So here is a carboxyl group, right? Carboxylic acid. Here's the amine. And most of them are going to be alpha amino. So what that means is that here's the carbonyl carbon. The first carbon next to it is going to be the alpha, right? So alpha amino. There are some bacteria that produce beta amino acids, but most of them are going to be alpha amino. Eukaryotes use only L amino acids. Some bacteria use D amino acids. And they're often found in bacterial cell walls and coatings and things like that. Most likely it's a way to stop enemy enzymes from attacking their cell walls and coatings. So here's glyceraldehyde. So in comparison, L-alanine, you can see it's the amino group that's on the left versus hydroxy group, right? So alanine has an R group of the methyl group right here. If the R group happens to be a glycine, glycine is an amino acid, but it doesn't have a chiral center. It's the only one that doesn't, because these two are the same. Then how we find them in nature under physiological conditions as they're what we call Zwitter ions. That means they have both positive and negative charges. So for example, here, why would that happen? Because at physiological pH, most carboxylic acids are ionized and most amino groups are protonated. And so because of the ionic attractions between this, you go to the health food store and you buy amino acids, they're usually white powders. And so adults need 8 essential amino acids, children need 10. And the significance of that is that 2 non-adult essential amino acids are synthesized by enzymes coded for by developmental genes. Okay. So as children get older, they develop the ability to synthesize 2 more amino acids, arginine and histidine. Okay. So those genes were there all the time, but they were turned off in children up until a certain age. So you should probably know the structure of these here: glycine, which is the easiest one, alanine, which is the second easiest one, then phenylalanine. You can imagine, here's alanine, I just put a phenyl group on it. Cysteine, lysine, and aspartic acid. If you know those, then you kind of get the idea about how amino acids are. So anyway, we take all amino acids, we put them into 4 different classes based upon their R groups, because whatever this is, all the rest is the same, right? From amino acid to amino acid. So I'm going to go through them from an organic chemistry standpoint. I don't know why someone's calling me. Sorry. I'm going to ignore it. Where is that thing I'm trying to find? Oh, here. Okay. So here's the amino acids. Here's the generalized structure. Here's that cellular pH. In other words, you get the Zwitter ion. And here's the shorthand method that they use. And so you have some that are nonpolar. So of the nonpolar, glycine is nonpolar. Now wait a minute. This whole thing should be ionic, but we're going just by the R group. What makes one amino acid different from the other is the R group. So the carbon to hydrogen bond is nonpolar. You can see for alanine, this is all nonpolar. And then look at the other ones. Valine, leucine, isoleucine, phenylalanine, because the benzene ring, when it attaches, is called the phenyl group. So this would be alanine. We put the phenyl group, so now it's called phenylalanine. Here's methionine. You can see that it's a sulfide, not a disulfide, but a sulfide. So electronegativity difference between sulfur and carbon is almost nothing, so that's a nonpolar bond. And proline is kind of unusual because the enzyme that produced this really took 1, 2, 3 in a row, which doesn't exist otherwise, right? And turned it into a secondary amine. Okay, so it's a secondary amine because it's bonded to 1, 2 other carbons. So you get this 5-membered ring. Then tryptophan has this aromatic ring on here. And I had to add that double bond right there because they made a mistake there. And if you look at requirements to be aromatic, we usually go by the Huckel rule. And so 2, 4 is not enough to satisfy the Huckel rule, but the 2 non-binding electrons on that nitrogen are involved as aromatic electrons over here, making it aromatic. So even though there's a nitrogen here, this is all aromatic. Okay, this nitrogen cannot act as a base because its electron pair is involved in making this aromatic. So one thing I will point out is that besides not having a propyl group with an R, because this is more like ethyl, and look at this one. This is like an isobutyl group, isn't it? And this is like a sec-butyl. So leucine and isoleucine, their difference really is just where that methyl group is. So you can imagine the enzymology required to synthesize these things. If you have valine, you need only 1 more enzyme to produce methionine. Okay. If you have alanine, you need only 1 more enzyme to produce phenylalanine. Well, why there's not a propyl group over here is kind of interesting. If you look at the difference between, say, alanine and valine, the only difference is a methylene group. Okay? 1 carbon difference. I'll bring that up later on because by the time we're looking at all the amino acids, what you're going to find is that there's not very many different functional groups. And then you're wondering, well, if there's not very many functional groups, there's not very much chemistry that's possible. But yet enzymes can do such amazing things. It's because of all the cofactors that are used with an enzyme. Cofactors are used to expand the capability, the chemical capability of enzymes beyond just what amino acids would allow.
Coenzymes.
I'm sorry?
Coenzymes.
Coenzymes. Yes. Okay, so those are the nonpolar. The polar ones are here. Notice that this is an alcohol. Okay, that's a primary alcohol. Here's a secondary alcohol. Here is phenylalanine with an alcohol. So if you already have phenylalanine, you need only 1 more enzyme to make it into tyrosine. Notice that this is a thiol, and they have it wrong in almost every textbook. You guys know that the difference in electronegativity here makes it a nonpolar bond. That should be classified as a nonpolar amino acid, but all the textbooks just copy each other, and that's the problem. Okay. So if you look at asparagine, what the heck is this? Here's an amide bond right here. Okay. So how would you make asparagine? Well, one way you could just take valine, you oxidize that terminal carbon to an aldehyde, and then maybe even a carboxylic acid, then you add ammonia to it. Make that simple amide. So we know this is polar. We know that the NH is polar. And look at the difference between asparagine and glutamine. There's really only the methylene group there. So getting back to what I was trying to explain to you about the limited number of functional groups in amino acids, why would nature say, well, let's make a whole different amino acid only different by one methylene group? Chemistry is exactly the same. So the spacing of things in proteins and enzymes must be extremely important. So besides nonpolar and polar, we also have acidic and basic. So notice that the two acidic amino acids are both carboxylic acids. And again, the only difference between them is a methylene group. So aspartic acid and glutamic acid. And then the ones that are basic. Oh, why are these be basic? So here's a ring, and why do they have that positive charge on there? Okay. It's because one of these nitrogens, and they can be written in different ways with bonds, double bonds in different places, one of them has non-binding electron pairs that can act to absorb a proton. And they showed that one right here. Okay. Then here's lysine, which is simply a primary amine. And then there's arginine. Now, what the heck is this functional group here? That's crazy. So here's a way for you to know the difference. So this has a nitrogen, that has a nitrogen. What do we know about amides? They're not basic, right? What about this one right here? It's also not basic. So we have a couple amino acids that have nitrogens in it that aren't basic. But look at the ones that are basic. This has 2 nitrogens. This has like 3. So an easy way to remember it, if it's got more than one, it's going to be more than one nitrogen, it's going to be basic. And you already know that a primary amine is going to be basic. And so while I'm telling you that the books are making a mistake, I found an article one time, this is published in 1991, where they measured the actual hydrophobicity, not just looking at electronegativity. And so you can see that cysteine really should be up here. Okay, it should be a nonpolar neutral amino acid. Okay. So one of my really good friends, he was much older than me, he passed away a couple of years ago, Professor Bhagavan. He wrote several books on biochemistry, medical biochemistry, and I was editing the last book that he wrote, and I said, let's change it. Let's fix where we put cysteine. And he thought about it, he knew I was right, but at the end he said, well, I don't want to rock the boat with the publisher, and he just didn't change it. Anyway, so questions so far? Okay. So now we need to understand what is electrophoresis. It's the movement of an ion in an electrical field. That's what electrophoresis is. isoelectric focusing is electrophoresis and a pH gradient. So the reason we need to understand that is we need to understand what is the isoelectric point. So the isoelectric point, which is designated as PI, is a pH where an amino acid or a protein has no net charge. And at that pH, there will only be zwitterions, and there will not be a net charge. So at the isoelectric point, at the isoelectric pH, there's no net charge on the molecule, and it will not move in an electrical field. So essentially the PI is just the mathematical average of all the pKa's in a molecule. And so this is a way to purify amino acids and proteins, because every single one's going to have its own PI. So if you look at acidic amino acids, they will have an acidic PI. If you look at basic amino acids, they will have a basic PI. So anyway, let's look at what happens with aspartic acid. Here, the pKa is 2.77. So at pH 2.77, you have no net charge on the molecule. So that means that this has to be charged, and these are going to be about 50-50 ionized and 50-50 protonated. So the net result is no resulting charge. So let's take this amino acid, and here's a pH gradient. So I just put 1, 5, 9, 13, right? So at pH 1, here's the concentration of hydrogen ions. Here's the concentration of hydroxide ions. At pH 13, here's the concentration of hydrogen ions and the concentration of hydroxide ions. And it's a gradient over there. So if I just put this amino acid in here, it's not going to do anything except diffuse. I won't get what I want until I actually put a charge across here. So here's the positive charge over here and the negative charge over here. So there's a voltage across this solution right here. So what would this molecule look like if I plopped it in at pH 1, where I have a whole bunch of hydrogen ions there? I'm essentially going to protonate everything. So if everything becomes protonated, the carboxyl groups have no charge. The only charge is on the amino group. But that positive charge is going to be repelled by that positive charge and attracted to that negative charge, so it's going to migrate. In other words, electrophorese in this direction. What if I take this same amino acid and I plop it over here? Here, I hardly have any hydrogen ions. I have a whole bunch of hydroxide ions. So I'm going to take the proton off of everything. So this is completely ionized. That's completely ionized. That has no hydrogen on it. Look at the charge on this. It's a -2. It's going to be repelled by that electrode, attracted to that electrode. So it's going to migrate in that direction. It's going to electrophorese in this direction until what happens? It gets to become neutral. So if I put it here, it's going to migrate that way. If I put it over here, it's going to migrate this way. It's going to focus at the pH, called the PI, where there's no net charge. So you're concentrating the amino acid at this place in the device. It's going to reach this point of saturation and start to precipitate here, which means that the solute-to-solute attractions are more important than the solute-to-solvent attractions. Does that make sense to you? So it precipitates right here. So if I had a mixture of amino acids, one of them might be over here, the other one might be over here, but this particular one's going to be right here. Okay. So something about glutamic acid I'll point out. Neutralization of glutamic acid with sodium hydroxide will give you sodium glutamate, or monosodium glutamate, MSG. So MSG gives the umami, or pleasant taste, as we talked about in chapter 27. It's one of the most abundant free amino acids in the body. That means you need it for something. What do you need it for? It's the most abundant neurotransmitter in the brain because it's the primary excitatory neurotransmitter. It's also the precursor to gamma aminobutyric acid, or GABA, which is the major inhibitory neurotransmitter. So green tea contains significant amounts of glutamate and threonine. So threonine is not an amino acid, but it's an amide form between glutamic acid and ethylamine. It also helps provide the umami taste and is known for its mood calming properties. So there's been controversy about MSG, especially put into Chinese foods at restaurants, and some people would get headaches from it and everything else. But is it like bad for you? Is it a poison? No, it's a natural thing that's in your body. But again, everything in moderation, right? So here is threonine, here's glutamic acid, here is gamma aminobutyric acid. Notice that all they did was to simply take off that carboxylic acid right there. So this is excitatory and this is inhibitory. They counteract each other. So here's some amino acids that can be dangerous. Here's the amino group, here's the acid. These are called hypoglycins, and they are toxins. They cause all kinds of problems. So it's found in the fruit of the unripe fruit of the seeds and seeds of this ackee plant. And relatives of that plant are lychee, longan, and rambutan. And we love those fruits, don't we? So the unripe fruit can contain enough hypoglycins to cause sickness. So in 2015, there was dozens of Indian children were killed, and they found out they were eating unripe lychee on empty stomachs. And that was what was killing them. Yeah, so they're enzyme inhibitors. Can you imagine why they're enzyme inhibitors? Look at this molecule right here. So you have this cyclohexane ring, which has all this ring string. And then in addition to that, you get a double bond over here. So you can imagine that this is extremely reactive. So if this gets bound to an enzyme, which is a protein, you put a little bit of stress on that ring, it's going to open up, and those carbons in the ring are going to be looking for new bonds to form. Well, they're going to form bonds with that protein. And that's going to permanently inhibit the enzyme or the protein. Anyway. So questions so far? Okay. I'm not telling you not to eat lychee, okay? Yeah. So let me find figure 2. So figure 2, I like this because it shows you all the different ways that protein structure fits together. So the primary structure of a protein is what? It's how the bonds are fit together. So here we have alanine and tyrosine. So notice that alanine has a methyl group here. Tyrosine has, the phenol ring over there, if you want to say that. Wouldn't that be different if these two were switched around? That'd be a different molecule, wouldn't it? So the sequence of amino acids in the chain is the primary structure. But the primary structure determines what the secondary structure will be. How is that? Well, here's hydrophobic. Here's hydrophobic. They're going to want to tend to be together. Here is polar. Here is very polar and very polar. Those are going to be water soluble, right? Here again is something that's non-polar again. So this, and this would tend to be together. So the primary sequence determines the secondary sequence. And there's really only 3 types of secondary structure. I know textbooks sometimes make it very complicated, but there's only 3 types. There's alpha helix, which they're trying to show here. There's beta pleated sheet. And then there's anything that isn't one of those 2, which we call random. So while the primary structure determines the secondary structure, the secondary structure determines the tertiary structure. How does that work? Because if all these amino acids right here were non-polar, and all these over here were polar, then this part would be outside of the protein. This part would be inside of the protein, away from water. So it directs how the protein will fold up into its 3-dimensional shape, which is known as the tertiary structure. In a tertiary structure, it's either going to be globular, which is spherical, or it's going to be more elongated, which those are the only 2 possibilities. And the tertiary structure determines what we call a quaternary structure. So for example, this is very similar to what you would see in hemoglobin. Hemoglobin is a spherical protein. It's made up of 4 spherical subunits that go together to form the hemoglobin. And that red disc right there is probably the heme iron they're talking about. So that means that this part of the protein is going to be attracted to that part of the protein. See how that works? And this part over here is going to be soluble in water. Same thing over here. So on the outside, these are going to be polar amino acids. On the inside, they may be nonpolar, causing them to want to clump together. Okay. That's not what I want, that one I want. Okay. So it's similar to what I put over here. So I'm going to expand it so you get a really good overall view of this. The primary structure of the DNA, the sequences of the base, are going to determine the primary structure of the amino acid. Primary structure determines secondary structure. Secondary structure determines tertiary. Tertiary determines quaternary. And that all determines the function. So how does the base sequence in DNA determine function? Well, it's because it directs the protein. So here's the plans. This is the machine, and this is what the machine does. So let's look a little bit closer at primary structure. And the peptide bond is an amide bond. Okay. So first of all, we need to understand that amides are not basic, because I take that non-bielectron pair in the nitrogen, I swing it into this double bond right here, then I chase that pi electrons back up onto the oxygen. Even though I have formal charges on here, all I'm doing here is I'm demonstrating that those electrons right there are not available to accept the hydrogen ion. So therefore, amides are not basic. In addition, if you look at the primary sequence of a chain, one thing you might think about is like, okay, would that be like a wet noodle? Would it be kind of like something much more stiff than that? And the answer would be actually quite rigid. Not very rigid, but rigid enough. Because of the partial double bond character right here, you've locked these guys into place. You do not have free rotation about that double bond right there. Okay? So that greatly limits the flexibility of an amino acid chain in a protein. So the peptide bond, or the amide bond, is formed by a dehydration, put together by an enzyme, obviously. And so the convention is, when we write it, we always put the amino group on the left. We call this the N-terminus, this the C-terminus. So when I do a dehydration and I form my amide bond, or the peptide bond, always the way we write it is the amino terminus, N-terminus on the left, C-terminus on the right. If you do it the other way, then you go to biochemistry jail. So what we put together here, we would call it alanylserine. It's like serine that's been modified with alanine. And here I switch these two around, and I would get serenylalanine. So I want to point out that different names, they're different molecules. Here, what do I have right next to the carbonyl carbon? This is an alcohol group. But over here, it's a methyl group. That makes it slightly electron withdrawing, making this a stronger acid. This is slight electron donating, making this a weaker acid. So you can see that the acidity of these two carboxyl groups is going to be different. So the sequence makes a difference. So the primary structure of a protein reflects the primary structure of DNA. It is not exactly the same. But in general, if a protein from one organism has a similar sequence as the same protein from a different organism, then we say that those two proteins have significant sequence homology. Similar primary structure. So for example, the insulin from pigs can be used to treat diabetes in humans because there's only one amino acid that's different. If you look at insulin in cows, there's 3 different amino acids. So that means that pigs are closer to humans than cows, according to insulin. Okay. So let's look at how insulin is produced. So the gene does not produce insulin. The gene produces messenger RNA in the nucleus. The messenger RNA is processed and matured, is worked on by different enzymes. Introns are removed, that we'll talk about later on. And what you end up with is proinsulin. Okay. And so then it has to go to the endoplasmic reticulum. The enzymes in the endoplasmic reticulum cut out this part right here. And now you have what's called proinsulin. And proinsulin has to go and get processed in the Golgi apparatus, where this blue part is taken off. And now we have insulin. It's two different polypeptides held together by disulfide bonds. And that is the active form of insulin. So if you are insulin deficient, that could be because of many, many reasons. It could be all the way back in the nucleus. It could also be in the endoplasmic reticulum or in the Golgi apparatus. Okay, let me see. Okay. So cytochrome C is part of the electron transport system in most organisms. There's been significant sequence homology across many organism types. That means the cytochrome C has not been altered very much by evolution. We say that it's a highly conserved protein. Yes, we're looking at proteins to understand evolution. So cytochrome C has been used to develop certain family trees like this. Okay, so here we are over here. Here's ginkgo, here's spinach. Yeah, so here's plants. Here's fungi. Here's insects. And so we can see that there is a split here between locusts and all these other things, insects in general. And then there was a split between the fly and the honeybee. And this is the amount of differences between amino acids in that one protein. So we can see that there's a split over here. So birds and reptiles went that way, mammals went this way. And then you can see that the rabbit went that way, the kangaroo went that way, chimpanzees went that way, monkeys went that way. Oh, where's humans? Well, we're over here. So we're more closely related to chimpanzees than we are to monkeys, and more closely related to monkeys than we are to, say, rabbits or kangaroos. So here's really what happened historically. People struggled to get the amino acid sequence of proteins. When I started graduate school in 1980, people would get PhDs on sequencing not even a whole protein, but just sequencing part of a protein. That'd be enough for a PhD. Nobody could sequence DNA back then. But then DNA became much easier to sequence. And here's the problem. If you take a protein and you cut it up into pieces that you could deal with, every piece is going to have completely different properties. Some might not even be soluble in any common solvent you can find. But for DNA, you cut it up into pieces, every piece has the same physical properties. So you can separate them on the basis of their mass quite easily in the same solvent. But this is what you would have to do to get the amino acid sequence of a protein. Purify the protein. Okay, for my PhD work, I purified a protein from a bacterial membrane. It took me, I would say, close to 4 years to do that. And then you have to separate the polypeptide chains, right, the subunits, and focus on one chain or subunit. Then you got to cut that into pieces that are big enough to actually do. And then you have to purify each polypeptide piece and then focus on one of those at a time. So how do you do that? You chemically remove each amino acid from the N terminus one at a time and you identify it. Then you have to go in the opposite direction, remove each amino acid from the C terminus one at a time, and then make sure you get the same sequence. And then you do this for all of the pieces that you have. But then you have to figure out, how do the pieces go together again? So to do that, you have to cut them in different places with a different protease, and then repeat the whole thing again. And then you're not even done, you got to go to the next subunit. So you essentially keep going until your grant runs out or you die. So now the definitions for what is a protein can be quite different. There seems to be this 50 amino acid cutoff. If it's 49 amino acids, it's not a protein, it's a polypeptide. If it's 50 amino acids, it's a protein. And I like to go by biological function. If that's the whole thing, if that's biologically functional, then that's a protein. Okay. Well, let's look at secondary structure. This is how the polypeptide chain kinks and folds. So 4 major types. Collagen, triple helix. I actually am putting that in a different section. We're going to treat that differently because collagen is a different kind of animal. It's a different category of protein altogether. So what I'm trying to explain to you is that there's only 3 types of secondary structure. There's alpha, which is alpha helix. There's beta, which is the pleated sheet. And there's anything that isn't one of those, we just call a random coil. And this will be specific to collagen. Okay. So let's see, find this again. Oh, okay. So here's kind of a view of alpha and beta structure. Okay. So here's an alpha helix right here, right? So what holds the alpha helix together? These are hydrogen bonds right here between the carbonyl oxygen of 1 and, where are we? And the amino group of another. Doesn't really show very well there. And so the forces of attraction are along the length of the helix, which means that I could probably just take that and pull it apart by this, and it's like a little spring, and you stretch the spring, and then when you let go, it goes back again. The forces that make it go back again are going to be hydrogen bonds. And then beta structure is a couple different ways, but they're attracted sideways to each other. Okay, so what's attracting them this way? Hydrogen bonds, but between one run and another run and another run over here. So beta pleated sheet, the attractive forces go in this direction. So it doesn't mean that you can take it and stretch it. It's not going to be stretchable. Excuse me. Okay. So we'll come back to the to the text in a second here. But here's a couple ways that beta structure can be affected. Here's head to tail, here's head to head, and here's also head to head. Okay. So there's different kinds of loops that can happen to get beta structure. And then before we get too far, let's look at the protein structure of an enzyme. Triose phosphate isomerase. Okay, well, you guys know what an isomer is, right? So what does an isomerase do? It turns one isomer into another isomer. So triose phosphate, you know what a triose is, right? Triose phosphate. So in other words, what would an isomerase do? Take the phosphate and put it on one carbon compared to another carbon. It moves the phosphate group around. Anyway, here's what I want to point out. These are sections of alpha helix, here and here. And how are they held together right here? By some random coil. Here are some sections of beta pleated sheet. How are they held together? Hydrogen bonding this way. So what do you have in the transition between beta structure and alpha structure? Random coil, because that's all there is. So you can see that this center of this donut looking thing is probably right where the triose phosphate gets bound. If we turn this on its side, you can see its structure from the side. So here's random coil, more over there, more over there. Okay. So anyway, let me get back to the, keep toggling all over the place because I'm not sure how to, there's got to be a better way to do it, but anyway. So going back to looking at alpha and beta. So the alpha helix is a twisted spiral coiled like the threads on a right-handed screw. Well, that's because all the amino acids in nature are L. So a really kind of cool experiment they did way back is they synthesized D amino acids and made a similar sequence to found with L amino acids. And what they found out is that, yeah, it still promoted alpha helix, but it gave a left-handed screw. So the reason that the alpha helix is a right-handed screw is simply because all the amino acids are L. And the R groups point out perpendicular to the structure. So larger R groups would repel one another, tend to prevent alpha helix structure. So it's really what's poking out of the alpha helix determines, is that section soluble in water or not soluble in water? The physical properties is what we're talking about. So it's held together by hydrogen bonding between the carbonyl oxygen and the amide nitrogen of another one, about 4 residues away. So that makes about 3.6 amino acids in each turn of the alpha helix. Okay. And in the beta pleated sheet, there's hydrogen bonding between the carbonyl oxygen of one section and the amide hydrogen of another section. And it's not stretchable. In random coil, is this anything that's not repetitive? Let's see. So what about tertiary structure? This is how a single polypeptide folds into a three-dimensional shape. And so you have globular proteins that are spherical. These are mostly water soluble. They're proteins like enzymes, hormones, antibodies, transport proteins, albumins. If you find it in the blood, it's going to be globular. Fibrous proteins are long and thin, usually water insoluble. So they're like structural or contractile proteins, like muscle proteins, sinews, collagens, things like that. And then finally, quaternary structure is how two or more subunits, which are polypeptides, fit together. So, anyway, there's only two basic possibilities for quaternary structure. They're either going to be monomers that consist of a single polypeptide, or they're going to be oligomers, more than one polypeptide. If they're an oligomer, they could be a dimer, trimer, tetramer. You get the idea, right? So, questions so far? So here's triose phosphate again. Okay. Here's myourethrin. Okay, so look at that. Alpha helix over here, random. I don't see any beta sheets in there at all. And what is this right here? It's a heme, right? That's a heme. Those are iron atoms, and these are the heme groups right there. Myo means muscle. That means heme. This must mean that it's in a erythrocyte. In other words, a red blood cell. So here we turned it on its side. Okay. So what do you think this molecule is meant to do? Find oxygen. Okay. Right there. So these are space-filling models, and these are ribbon models. So you can see the space-filling models are good for some things, but they're kind of hard to figure out. You cannot see secondary structure in here at all, okay? But it's these space-filling models that are used in pharmacy. There's lots of computer programs now where they actually get the structure, the three-dimensional structure of the protein. Here's where the natural agonist binds. When I'm trying to find an antagonist to act as a drug, it also has to fit in here. So they can use all kinds of modeling to come up with potential molecules that they could then synthesize and test to see if it actually does work with that particular protein or enzyme. So ribonuclease is an enzyme that breaks down messenger RNA. So you can see it probably lays right across here, lays probably right across there, and it cuts it. So this figure right here tries to identify all the different forces that holds proteins together into its viable functional state. So what they're showing over here in the alpha helix is hydrogen bonds. Here's some random, and then for some reason they did this instead of making it look like it's attractive forces. Those are simply hydrogen bonds holding the sheet structure together, the beta structure together. Here's some more random coil. And here's what's called metal ion coordination. What the heck is that? So this negative charge is attracted to that positive charge. That negative charge is attracted to that positive charge. So this forms an attractive force between this part of the protein and this part of the protein to hold it together. So in this case, they're using magnesium. It could be really any metal ion that's particular for that protein. But what would happen if you didn't have that magnesium ion right there? If you just yanked out that magnesium right there, now you would have a negative charge and a negative charge. That would be repulsion. You see how important these metal ions can be? It really turns, in this case, repulsion into attraction. Here is a region where you have a whole bunch of hydrophobic amino acids over here. So these are going to be soluble in themselves, okay? Especially if you have hydrophilic ones out here in contact with water. Here's a disulfide bond. Chemically, there's a covalent bond holding these two parts together right there. And then they're just showing you side chain hydrogen bonding between an alcohol and some carboxylic acid group. And then we also have electrostatic attraction. If this is ionized and that's ionized, that's an attractive force. Okay. So could the pH have an effect upon the structure of a protein? Absolutely. If I were to protonate this with low pH, then I don't have this ionic attraction anymore. Or it's much weaker, I should say. Okay, so to go over them a little bit more, hydrogen bonding, you guys know what that is. Hydrophobic, hydrophilic interactions. That's why if you look at a protein that's a water-soluble protein, inside the protein, away from water is where you're going to find the hydrophobic amino acids. On the surface of the protein, you're going to have the hydrophilic ones. Okay. Then salt bridges are another one. Okay, so why do we not call this an ionic bond? It is an ionic attraction, but it's not as strong as an ionic bond. Like here's an ionic bond, right, between sodium and chloride. But if I put sodium chloride in water, I get this hydrated sphere and this hydrated sphere. So the attraction is much less, but it could still be an attraction. Here's another example of metal ion coordination. So why do we need cobalt as a trace mineral? There you go, right there. And finally, we have disulfide bonds between the amino acids that have a thiol in it, the cysteines. Okay. So here's two thiols. You oxidize them, you get a disulfide bond. One of my friends from high school, she had kids the same age as my kids, so we would sometimes meet and take all the kids down to the beach. And anyway, it's like, okay, I'm going in the water with the kids, and oh, I can't go in the water because I just had my hair done. I'm like, what do you mean? Oh, it's a permanent. So first of all, I really didn't know why a Japanese lady would want to get a permanent to make your hair curly. That was already weird to me, but And I told her, I was like, well, I guess you didn't really get a permanent, did you? You went to the beauty parlor and you got a temporary. Then I started harassing her about the beauty parlor anyway. It's like, what kind of people do you see in the beauty parlor? Beautiful people? Not necessarily. So I told her, yeah, she went to the ugly parlor to get a temporary. So when they give you a permanent, it smells, right? If you walk by one of those parlors, it really stinks. Because what they're doing is they're taking the disulfide bonds in the hair, reducing them to thiols, and then putting the hair in a different shape, bending the hair around, and then reforming disulfide bonds to try to keep it there. But as we learned about disulfides, it's easily reversible. Okay. So here's an example of the importance of attractive forces in protein function. About 100,000 Americans have sickle cell disease, and the red blood cells become sickled. So here's a normal red blood cell. Here's a sickle cell at full maturity. And in fact, people don't realize this. If you look at the shape and dynamics of a red blood cell, it is perfectly designed to pass through the smallest capillaries. It's like a Surtz looking thing, but it's very flexible, very easy to shape it into different forms. But because of the disc type structure on both sides, there literally is no way you can turn it where it doesn't catch the flow of water. It's an amazing design. Anyway, they don't clog up in capillaries, but the sickle cells do. So that's exactly what's happening right here. So the problem with people with sickle cell anemia is that it's a circulatory problem. They lose blood circulation to parts of their body. Okay, that's what ischemia is, means blockage. Okay, so reduce blood flow, lower oxygenation of tissues, and normal red blood cells live about 120 days. Sickle cells only, 2 weeks or so. So how do you fix that? Bone marrow transplant. What are you doing with the bone marrow transplant? Well, that's where all the blood cells come from, is from your bone marrow, right? So what you're trying to do is replace the gene that codes for this with the gene that doesn't code for that. And what is the difference? In sickle cell anemia, the two beta chains of hemoglobin have a single difference in primary structure. A glutamic acid is replaced with a valine. If you replace a glutamic acid with an aspartic acid, most likely you'd have still a functional protein. But in this case, you're taking a polar and acidic amino acid and replacing it with a non-polar one. So that causes a change in the shape of the hemoglobin. And when you pack all those misformed, misshaped hemoglobin molecules into a cell, it makes the cell change shape. Okay. A single mutation is called a point mutation, where one amino acid is different. So if this shortens the life of people, why is this gene still around? Wouldn't you think that that would make people live shorter and die quicker and have all kinds of diseases? The answer is yes. But the reason why it persisted so long is because it gives added protection against malaria. And at least that person lives long enough to get through the reproductive age to pass their genes on. So here's some terminology. So hydrolysis means hydrolysis of the amide bond that disrupts the primary structure. That's catalyzed by enzymes or by acidic or basic conditions. You hydrolyze proteins in your diet, first with the acidic conditions of your stomach, and then under neutral conditions in your intestines is hydrolyzed by enzymes. Whereas denaturation, also known as coagulation, is simply the loss of biological activity due to alteration of secondary, tertiary, or quaternary structure. You haven't hydrolyzed it. Okay? Some proteins will renature once the agent or the treatment is taken away. So RNase, if you've ever worked with RNA, you'll find that RNase is simply everywhere in the environment because of bacteria and mold and everything else. It's really difficult to get rid of. You could put it in the dishwasher, and while it's under the conditions of the dishwasher, it's totally denatured. And you take it out of the dishwasher, then you let it dry, and guess what? That enzyme reassembles. Simply because its primary structure directs secondary, tertiary, and quaternary structure. So what kind of treatments cause denaturation? Heat. Okay. Why would heat do that? Because, simply if you heat something up, things bounce around more. You're breaking attractive forces. Okay. Why do we cook our food? Because we denature the proteins, and that makes them easier to digest. UV light and microwave radiation, why? Because all that is absorbed by the proteins and turned into heat. Organic solvents. For example, if you take a water-soluble protein, the amino acids outside in contact with water are going to be polar. Now you put it into a non-polar solvent, you basically tell the protein to turn its inside out. Violent agitation, similar to heat, but also similar to organic solvents. Because if you take a protein solution and you shake it up, there will be bubbles there. Okay, so what are those bubbles doing? Well, inside the bubble, outside the bubble is air. Air is a non-polar solvent. So again, you're basically trying to turn the protein inside out. Soaps and detergents emulsify the protein by disrupting those hydrophobic and hydrophilic forces, similar to what organic solvents do. pH changes, that affects protonation of acids and bases, can make charges appear or disappear, affects salt bridges. Heavy metal ions form salt bridges with carboxylic acids. So they can replace some of the natural salt bridges in there. The problem with the heavy metals is that, okay, so now the protein isn't working good. So you destroy the protein, you cut it all up with proteases and stuff like that. And then what happens to the mercury or the lead? It's still there to go ruin another protein. And finally, redox agents can affect disulfide bonds.
So at what structure would you say a protein is no longer biologically functional? Is it like at the second, third, first?
It could be quaternary, tertiary, or secondary.
So it just depends on the protein then?
Yeah, sure. Yeah, they're all different.
Okay, I see.
Mhm. So how do we classify proteins? Well, one is by their functions. Okay, so we got enzymes or catalase, transport proteins such as hemoglobin, lipoproteins, membrane transport proteins such as the glucose transporter that brings glucose in and out of the cell, contractile proteins, we have structural proteins, we have defense proteins, we have regulatory proteins, receptor proteins, nutrient proteins. Like if you look at casein of milk and ovalbumin in eggs, C proteins, the only reason they're there is to provide a source of amino acids to the baby cow or the growing egg or the growing plant. Another way to classify is by composition. So you have simple proteins. That means they have only amino acids in them. And then you have conjugated proteins. Those contain a compound other than an amino acid. It could be an iron atom. It could be the heme group. And of conjugated proteins, we often use the word prosthetic group. A prosthetic group is something that is covalently bonded to the polypeptide. It could be a vitamin, a phosphate, carbohydrate, lipid, nucleic acid. It could be almost anything. So for example, if I take hemoglobin and I boil it, so I disrupt quaternary, tertiary, secondary structure, right? The heme is still covalently bonded to the amino acids. So the heme is considered a prosthetic group, not just a cofactor, but a prosthetic group. And then we could categorize them by shape. They're either globular or fibrous. We could classify them by the number of subunits or by where we find them. I had to go through the literature for my graduate work. I had to go through the literature, the older literature from the 50s and 60s and stuff like that. And I'm trying to figure out what they're actually working on. And they would tell you they're working on this particular enzyme, but they didn't tell you where they got it from. So maybe they might tell you it's from a cow. Okay, but what part of the cow? Anyway, so the more information, the better, right? So what we're going to do now is just look at some proteins and look at some of their functions. It's kind of like a survey. So vasopressin is a nonapeptide. What does that mean? Nona is 9, so there's 9 amino acids in there. Has 1 disulfide bond. Its mass is about 1,000. It's molar mass. And what does it do? It's a hormone that promotes the absorption of water into the kidneys and raises blood pressure. Okay. Oxytocin is also a nonapeptide. It promotes the contraction of smooth muscle. They use it to induce labor and childbirth. It's also used as a neurotransmitter in some specialized nerves, and it differs from vasopressin by only 2 amino acids. So yeah, I would call these proteins because they're biologically functional. So insulin is a polypeptide hormone released by the beta pancreatic cells and tells cells to uptake glucose from the blood and basically to store it as glycogen. It's got 51 amino acids in a molar mass of about 5.8 thousand. So glucagon, another peptide hormone with 29 amino acids, mostly in alpha helix structure, is secreted by the alpha pancreatic cells. It's proglucagon. Well, as proglucagon, it has to be converted into active hormone. So it acts as anti-insulin. So it promotes the release of glucose into the blood from glycogen. So the levels of glucagon release are fairly constant. The reason is you never want your body to be have your blood be hypoglycemic. You don't want that to ever happen. So how you control your blood sugar is more with the variable release of insulin than the steady release of glucagon. But both of those come from the pancreas. So the pancreas pretty much controls blood sugar. So cytochrome C we talked about before. It carries electrons in the electron transport chain using a heme prosthetic group. About amino acids, 100 amino acids long. It's a monomer. Mass of about 12,000, making it barely able to exit the mitochondria through the porin opening in the outer mitochondrial membrane. And we've learned that if you start to find cytochrome C outside the mitochondria, that's an early indication of apoptosis, also known as programmed cell death. So when your mitochondria start to be destroyed, you got no way to make energy, you're going to die. So keratins make up hair, fingernails, skin, wool, the horns of animals. They're fibrous proteins, mostly alpha helix, so they're stretchable. So they're extracellular proteins. That means that they're made outside the cell. There's usually two alpha helical links twisted together into a dimer, and then the dimers are twisted into a tetramer. And the tetramers are then used to build the biological structures. So, here's a cross-section of a hair. Okay. So here's the alpha helix over here. Here's the dimer, right? And then all the dimers are put together into a filament. And these are the cells right here. And they make these outside the cell. Okay. So here's the two chains making a single coil, and then you have a protofilament where they start hooking together. And then they would be pretty limp. So how you stiffen that up is with, what's the word, cross-fixing. I can't think of the word right this second. Where are we? Yeah, using disulfide bonds, okay? So fibroin, which is referred to as silk, is a fibrous protein made by insects and spiders. It's stronger than steel, stronger than Kevlar. It's mostly anti-parallel beta sheets, that means head to tail, beta sheets, and it's not stretchable, even though there's no cross-linking. So hair is stretchable. Why? because of the alpha helix structure. Silk is not stretchable. Why? Because it's beta. Yeah, so they're trying to make synthetic spider silk proteins. Hopefully they'll be able to do that. It costs a lot of money in terms of fossil fuels to make modern clothing. Nylon and plastics, all the monomers come from what? Fossil fuels. Cotton is probably better, but then it costs all kinds of energy to make it into cloth, right? It's all done by fossil fuels. I have friends that sometimes lament about, oh, you know, if we could only go back to the old days, you know, everything was so much better then. It's like, you're pretty much clueless. If we were to take fossil fuels away from you, you would be simply living as a caveman. Simple as that. So tubulin, question? So tubulin is what makes up microtubules of the cytoskeleton. It's a globular protein that polymerizes into tubes in response to an increased ionic environment. Tubulin makes up centrioles, spindle fibers that separate chromosomes before cell division. And I just added this one part. It's something that we've talked about, but it's always usually outside of a regular textbook, but intrinsically disordered proteins. These are proteins that do not keep a specific shape, but they change shape upon binding to other proteins to affect some biological process. And I just put it in here just because I wanted to have it in here to eventually connect it to some biological function, but we know about them. We don't understand exactly how they work. Then we get to connective tissue. So these are synthesized outside the cell by fibroblasts. So what are they? Those are a type of cell that produces extracellular proteins that are fibers. So then we have chondroblasts that are specialized to work on cartilage and osteoblasts that are specialized to work on bones. But the connective tissues contain mostly elastin and collagen. Okay? And they have cross-linking between them. So what are the elastins? Well, those make up ligaments, skin, lungs, arteries. These are all things that have to be able to be stretched. In other words, they're elastic. So the major structural secondary structure is random coil, but they do contain both fibrous and amorphous components. So that's why I put this intrinsically disordered proteins statement there somehow. So you get this rubbery interconnected network. Yeah, a couple other things about them. So by the way, connective tissue in terms of nutrition are not very nutritious. So when you're eating a piece of chicken, you know, the regular white or brown meat is easy to take off, easy to eat, but the part that's hard, you have to keep chewing it and it's crunchy and all that kind of stuff. It doesn't give you much nutrition anyway, because it's not very many different kinds of amino acids. Very limited is the number of kinds of amino acids in it. So collagens. So I made this just to try to get you to understand why I took that particular structure out of the three major types of tertiary structure, which are alpha, beta, and simply random. So here's the triple helix of collagen, okay? It's a right-handed triple helix. The tertiary structure is that it's fibrous. The secondary structure is a left-handed helix, unique to collagen. It doesn't exist without having the tertiary and quaternary structure intact. So in other words, if I took this apart, I don't see this left-handed helix. Okay, so it's not a normal part of a tertiary secondary structure. It only happens when it's folded up into the biological structure of collagen, and it is unique to collagen. So the primary structure contains a lot of glycine, proline, and hydroxyproline. So what's unique about glycine is our group is simply the hydrogen. And the hydrogen takes up almost no space. So to take it and turn this thing into a kink when there's a bunch of hydrogens there is not a problem. And what about proline? Proline is that one amino acid where the three carbons, the propyl group, got turned into a new bond with the amino group. So in other words, if you have a whole bunch of prolines in a section of a protein, it will cause it to turn. And then what the heck is hydroxyproline? That's proline where another enzyme came along and put a hydroxyl group on it. So it's not one of the 20 normal amino acids. It's found quite a bit in collagen, but only in collagen. So that properties allows it to kink up quite a bit when you put it into this tertiary and quaternary structure. So how do you get that into fibrils? Okay, so here's the formation of what's called tropocollagen. So here's all the amino groups over here, the C-terminus over here. Here's your triple helix, and here your collagen triple helix. An enzyme comes along and cuts those off, right? And then those tropocollagens are associated into the collagen fiber, okay? And then you have these cross-links going on like this. So here's what the cross-links are looking like, right? Then you assemble them all into this fibril. So if you really think about it, it's not that much unlike rope. Okay. So you have all these little microfibers all trying to do the same thing. They make this fibril right here. Then if you look at connective tissue is like you have some fibrils going this way and others going this way. You make this two-dimensional structure that is extremely hard. It's a little bit stretchy, but extremely strong. Oh. Okay, so yeah, we better finish. I just noticed it's 3 o'clock already. So older vertebrates, such as myself, have more disulfide cross-linking in their collagen, so their meat is tougher. Vitamin C is required for that cross-linking. Without vitamin C, you get scurvy. Scurvy is a connective tissue disease. Anyway, so here's a little bit of information about the Achilles tendon, just to give you an idea. The half-life of collagens is very long, about 3 years. So if you're a professional athlete and you get a tendon injury, more likely you're not going to wait like 3 to 6 years for it to heal properly. You're going to get some surgery on it. Anyway, we'll stop right there and we'll continue this on Friday. Anybody have any questions before we stop? Are you getting your money's worth out of this class? I think so. Okay, so I will send out an announcement today about the exam 2. And yeah, if you have any questions, just email or yeah, put it up in the forums, okay?
Or if you have time, do you have time to go over the phospholipid from chapter 28?
Sure. Okay, so what I'll do is I'll stop the video and then we'll just go over that. This means I have to get my iPad going here.
I think you just turned off your video.
Yeah, I turned off the video, right?
It's still recording, but I mean, that's okay.
Oh, I see what you're saying. Okay. Start video. Where's the recording? Oh.